So in this video, we're going to talk about protein. When we hear about the word protein, the first thing that comes to mind is like, oh, muscles, right? It's like you need to eat lots of protein, meats rich in proteins, eggs rich in proteins, and it's gonna help you build muscles and make you look lean and strong. Well, all these things are true, but in the biological context, proteins are actually much more than just helping you build muscles. In fact, if you don't have protein, you're not going to survive. It's actually the most important biomolecules that determine your basic characteristics. So what are proteins? Proteins are organic compounds containing these basic elements, which are carbon, hydrogen, oxygen, nitrogen, and sulfur. If you look at it, right? Carbon, hydrogen, oxygen, nitrogen, sulfur. But not all the building blocks of protein have sulfur, but most of them have sulfur. So that's kind of like the reason why it gives protein a little bit of distinctive smelly smell. They play a lot of roles in the body, play crucial roles in transporting, signaling, catalyzing, storing, defending, enabling movement in cells. And throughout the whole curriculum, we're going to see protein show up often because they basically do all the things that makes a living cells survive. So the monomer of protein is actually amino acid. And the picture below shows the molecular structure of an amino acid. And you have 20 different types of amino acid. If you look carefully, it's made up of different groups attached to the central carbon. To the left is an amino group. And then you have the hydrogen group, the carboxyl group, and the random group, R group. And those 20 amino acids have the same amino group, carboxyl group, and hydrogen group, but they differ in terms of their R group. So the R group is also called the variable side chain. It determines the amino acids properties. 20 of them, they all look very different from one another. Kind of similar from one another. And you can group them into 4 different categories, depends on their properties. The first one is non-polar side chains, hydrophobic. And then we have polar side chains, which are slightly hydrophilic. Then we have the charged one, the acidic amino acid and the basic amino acid. Acidic amino acids are negatively charged, whereas basic amino acids are positively charged. Key. So amino acids have functional groups that can attach with one another to remove one water molecules in order to attach them together. So this process, as we have learned in previous videos, this process is called dehydration synthesis. By doing this process, right, you'll be able to form what we call a peptide bond. So if you have multiple amino acids, not too much, you're gonna form a short chain of amino acid called a peptide, or a longer chain of amino acid called polypeptide. So a peptide bond is these connections between the 2 amino acids after the process of dehydration synthesis. And this process is going to release a molecule of water. Proteins can differ in the numbers and sequence of amino acids. You can have very short chain of amino acids that have its own unique functions, or you can have a protein that has like about 10,000 amino acids and it's still going to work. So if you chain out together, change all these amino acids together, you're gonna see that they're gonna have 2 very different ends. The one that starts with amino group is what we call the N-terminal. And then the one that ends in the carboxyl group is what we call the C-terminal. The N-terminal is kind of like the starting point of the amino acid chain. And then the C-terminal is kind of like the ending part of the amino acid chain. To Together, we have this polypeptide chain that has different R groups, and they can have different properties, right? Remember, you can have a hydrophobic, hydrophilic, negatively charged, or positively charged R group. So protein structures can actually be defined by four levels. They are primary, secondary, tertiary, and quaternary structure. And we're going to go through them one by one. So primary structure is basically the amino acid sequence of the protein. Remember, you had 20 different types of amino acids. So if you start from beginning, from the the N terminus of the polypeptide, you're going to have different types of amino acid chain together. So this sequence is what we call primary structure. Then those repeating units of amino acid are going to have the carboxyl group and amino group being repeated, right? And they are all electronegatively charged. It means they're slightly polar. So you're going to have constant partial negative and partial positive charge from those terminals. And no, sorry, not from those terminals, from those group. And because of this repeating partial positive, partial negative, they're going to form hydrogen bond. Remember, hydrogen bond is the attraction between partial positive and partial negative. So right here, those dotted line represents hydrogen bond. And you have two ways of forming hydrogen bonds within the protein structure. If it coils around, is what we call an alpha helix. If it folds to kind of feel like a paper sheet and then you fold it in half, that's what we call a beta pleated sheet. But regardless of whether you coil for, the idea is the same. It's the attractions between the repeating units of amino group and carboxyl group. Then we zoom in more, right? Because of this hydrogen bond formations, you're allowing those R group to be very close with one another. So those R group are going to form their own unique attraction as well. And as a result, it's going to determine the overall 3D shape of the polypeptide. And it results from the interactions between the R groups of the amino acids. So the difference between secondary structure and tertiary structure is that in secondary structure, it is mainly hydrogen bond, and it has to be between the repeating amino group and carboxyl group. Two types of folding, alpha and beta. The shapes is not that unique yet. But tertiary structure is this very unique structure. It contributes to the 3D shape of the protein, and it's the attractions between the R group. Then we will have quaternary structure. Quaternary structure results when two or more polypeptides come together to form one individual big protein structure. So let's dive deeper into the R group. So you can see that all these boxes are different types of R groups. The color is representing the different types of property. So let's start with yellow group. So the yellow group, there are hydrophobic, non-polar R group. Because they cannot dissolve in water, they're gonna come stick with one another. This process is called hydrophobic interaction. It's kind of like how water is like pushing all the oils, and all these oils are like hugging with one another because they just cannot dissolve with water. So this process is hydrophobic interaction. Next, we'll have the polar group here. So you can see that this OH is, the H here is partial positive, whereas this O is partial negative. The dotted line represents hydrogen bond. Next, we have a very specific type of R group, and this R group contains sulfur. This amino acid is called cysteine. The only amino acid that contains sulfur. So when two sulfurs come together, they're going to form a covalent bond. And last, we'll have a negative charge R group come closer and attracted by a positive charge R group. And this attraction is ionic bond. So you can see that because of the properties of the R group, there's so many different types of R groups. and depends on the sequence of the primary structure, is you're able to fall into multiple different shapes, right? So this gives rise to many different many unique structure of the protein. Then for quaternary structures, right, some examples include first collagen. You often heard about collagen as this soft tissue that's between the bones. So this is a fibrous protein that consists of 3 polypeptides coiled like a rope. Then we have another type of protein, which is also a quaternary structure. This is hemoglobin, which is found in blood cells. So hemoglobin is made up of 4 polypeptides. We call it 2 alpha subunits and 2 beta subunits. So in summary, right, we can see that the shape of the protein is really affecting its function. And by changing the sequence of the amino acids, you're essentially changing the shape of the protein, right? Because the way how you arrange the amino acids is going to be different. And they're going to form different types of bonds. So that's why it's very important to understand this. So in summary, proteins contains carbon, hydrogen, oxygen, nitrogen, sulfur. They play crucial roles in transporting, signaling, catalyzing, storing, defending, enabling movement in cells. They are monomers amino acids. There are 4 types of structures. Primary, secondary, tertiary, and quaternary. Depends on the interactions that they form with one another. And protein can differ in the number and sequence of amino acid that leads to different shapes and chemical properties. That's it for protein, and I'll see you in the next video.